There are potentially thousands of secreted factors circulating in your blood. For a huge fraction of them, we have no idea what they bind to. That gap, a ligand with no receptor, is one of the bottlenecks in physiology. Today, two people who've thought hard about how to solve that problem. Welcome to EXO Chats, the podcast of EXO Beyond the Cell, the journal from Science Exploration Press, dedicated to everything happening outside the cell. I'm Brent Stockwell, editor-in-chief. In each episode, we sit down with researchers who are drawing the maps of the world that cells live in. If you want to read the papers that we discuss, they're all freely available at the website EXO Beyond the Cell. Secreted proteins are how cells and tissues talk to each other. Hormones, cytokines, growth factors, peptides, and metabolites move between cells and organs to keep the body functioning and integrated. Cataloging these factors and knowing what they do are two very different things. The limiting step is always the same question: what's the receptor? Our guests today have written the definitive map of how we attack that problem. Norbert Perrimon is a professor of genetics at Harvard Medical School and an HHMI investigator, and his lab has spent years building the tools and the biology of inter-organ communication. And Dr. Myeonghoon Han is the first author of this review and a member of Professor Perrimon's lab. Professor Perrimon and Dr. Han, welcome.
Thank you. Glad to be here.
Thank you for having us.
Great. Okay, so let me start with the big picture. So the catalog of circulating factors has really grown enormously, but the receptor annotation has lagged behind. For our listeners who don't live in this world that you're in, how big is that gap? And why is closing that gap important for what we want to do downstream and understand these secreted factors? So perhaps we'll start with Professor Perrimon.
Yes, this is really a fascinating question. I mean, the work in our lab and many other labs in the field have identified a number of really important secreted proteins that are acting across tissues and cells. And we have a very good understanding about the function of a number of them, but there is really a major knowledge gap in terms of the complexity of the secretome and the receptor that they bind to. So just to give you a sense, if you, like in Drosophila, if you just estimate computationally the number of the complexity of that secretome, the various estimates are about maybe 2,500 or so potentially secreted factors. And but those are computational predictions. We don't really know exactly how many of those are real. And our lab has actually performed a number of experiments to try to identify the secretome using a number of studies using proximity labeling, where we simply, for example, expressed a biotin ligase, which is going to biotinylate all the secreted factors. We accomplish this by expressing an enzyme called turbo-ID or BIR-A and so on in the endoplasmic reticulum, and then such that all the secreted factors become biotinylated. And then we can use mass spectrometry to identify the biotinylated components in the blood, for example. So using an approach like this, we've identified, we had a recent paper where we identified about 535 actually secreted factors. So that's in flight. So there is about a number of hundreds of proteins which have been validated as being secreted, and there is probably 1,000 which are predicted. So that's about the scope of the secretome in the fly. And interestingly enough, in humans, in mammalian systems, the numbers is actually quite similar. So if you do computational prediction, I think the number is about like between 3,000 to 5,000 potentially secreted factors. And then again, there's been some extensive proteomics experiments performed which have identified about 700 or so secreted proteins. So the secretome in flies and humans is about in the order of, let's say, 500 to 700. And what we need to understand is what the function of those secreted proteins are. And so for some of those, we know very well what they do. And this is really, I mean, the result of many in the field. Like, for example, you have factors like the Wnt proteins, the edge-joint proteins, TGF-betas, and so on, where we know very well how they act. The receptors are known, their signaling pathways are known. But for many of the others, of those newly discovered proteins, we don't really know what the receptors are. So it's really very important to identify the receptors of those orphan ligands to be able to characterize their function. And this is really where what we've been trying to address is to develop methods which would allow us to identify the receptors of those putative secreted proteins or orphan ligands. And so this is what really inspired this review. And Un in the lab is a postdoc in the lab, which has been really taking the lead on this. And it actually fits. It's a review that we decided to write because Un is trying to develop methods to allow us to identify the receptors of those secreted ligands. And over the past few years, he's been going through a number of different approaches. And this is a very challenging problem. I mean, as he can explain a little bit further, there are methods which allow you biochemical approaches, which can allow you to identify maybe the receptor of one or a few ligands during a postdoc time of 3, 4, 5, 4 years or so. But the challenge when Un joined the lab was to try to see if he could develop a method that is scalable, where we could identify, let's say, the receptors of maybe 50 or so putative secreted proteins. So this is a very ambitious project that he started, and he's made some progress on this. But here, the idea of this review was that since he has spent so much time trying to evaluate all the different approaches, was really to put this work in the context of the body of knowledge of the different approaches. So that would also help him to present better his work. And we saw it as an opportunity because it was really not such a review out there previously.
Yeah, that's a perfect foundation, I think, for the details of what's in the review and these new methods to do this kind of mapping systematically. So let me follow up on that. Dr. Han, there's a bit of a paradox in my view, and as I'm reading the paper. So the properties that make the interactions between these secreted proteins and their receptors interesting in that they could be transient or weak or tunable, also make them hard to capture systematically if we think about how to do that. So can you explain to the listeners more about that point of why it's difficult to identify receptors for ligands using affinity purification, mass spectrometry, for example? Where is that successful and where is that difficult?
Yeah, that's quite important point in this review because so like everybody knows that the affinity pull down has been extensively used to explore the protein-protein interaction, not only just the extracellular protein-protein interaction or just but also the intracellular as well. But the tricky part of this affinity pull down is we need to optimize the binding condition to preserve the protein-protein interaction of our interest. But the most interesting characteristics of the extracellular protein-protein interaction, it could be the transient, and also somehow it could be weak because it has wide range of the binding affinity from the nanomolar to the micromolar range. So because instead of there's in absence of the knowledge of the ligand receptor pair, it is really tricky to get the optimal binding condition, and also we need to prepare the certain physiological condition that ligand and bind the corresponding receptor exist at the same time. So that's why, because of these current limitations, we was started thinking about the new approaches.
And let me follow up on that because one of the points you made in the review is that glycosylation is often important in terms of receptor-ligand interactions. And so tell me a little more about that. I think a lot of people don't think about that. So where is that helpful and where is that making it more difficult to identify these receptors?
Oh, yeah, sure. So another interesting feature of the secure proteins or extracellular proteins that they undergo the post-translational modifications such as the glycosylations and disulfide bond formation and also the palmitoylations. So because of these diverse post-translational modifications, um, we it's difficult to predict the protein structures of it. And also, I mentioned that part in the AI based approaches. So current AI based approaches only allow us to input the FASTA sequence, the amino acid sequence. But more advanced version, they allow us to submit the glycosylation as well, but the patterns of the glycosylation and the combination of the post-translational modification is quite diverse and very complex. So it's practically hard to trace with the current in silico prediction tools. So it's quite challenging. So we need to prepare in a actual biological samples that resembles to the in real biology. So that's the caveat we need to think about.
Do you think computation will get there one day, or is this something where experiment is always reigns supreme? You just have to do the biochemistry and figure that out.
That's really interesting questions. I don't know. Actually, the development of the AI era is really so quickly and so fast. So someday, maybe a lot of scientists already jump into these AI based prediction programs. So someday, maybe we can use that tool to cover the every other combinations. But at this moment, when I write this review paper, I think the best way is to combine the AI tools and the biochemical approaches at the same time. I think that's what I can say.
Okay. Oh, Professor Perrimon, go ahead.
Yeah, in regard to the AI and the PTM, I think it's not clear at all how well this is going to work. I mean, if you just think about like glycosylation with the glycosaminoglycan chain, right? The lengths of the guide chains can be very variable. So I'm not sure how those AI models would account for the variability because we don't even know actually in vivo what is the actual length of those gap chains. So I think this is a very concerning issue. I think because you may be able to do some modeling with some type of PTM, but for specific ones, which are again where there is a lot of variability, this may not be so simple.
I think it's a great point. People forget that everything is not DNA and protein. There are other things that are important, like sugars and lipids, that are hugely complex, and still we need the basic biochemistry. So let me turn to some of the other approaches you talked about for mapping these ligand receptor interactions in the paper. So Professor Perrimon, you built your lab in many ways on the sort of genetic screening and model organisms and the power that that can bring to understand physiology and inter-organ communication. So how is that going to be useful, or how has it been useful for ligand receptor discovery? Can you bring the genetic tools into the picture here?
Well, I think I mean, some of the power of the genetics is to really validate some of those biochemical interactions, right? And so one type of experiments we can do in flies, let's say if we have a putative receptor identified by some kind of binding assay on either biochemical or some other type of approaches that we discuss in the review. What you can do going back into an in vivo system is that you can generate situations where if you think that a specific ligand is coming from a tissue A and is acting on tissue B, right? If you might expect to see the same phenotype if you overexpress the ligand or if you activate the pathway downstream of the receptors. And then in flies, what we can do quite easily are those genetic epistatic experiments where, for example, we could suppress the effect of an overexpression of a ligand by removing the receptor in the target tissue. So I think the two really go together. And I think for us, the approach that we are trying to take is to try to do either a biochemical screen or a CRISPR type activation screen that Un described, I think, in the review. And then to eventually make a set of predictions and then go back directly in vivo and use the genetics tool that we have to validate precisely this interaction.
How promiscuous, as I was thinking about that reading the review, I thought there's one issue of like promiscuity of receptor ligand interactions. Like one receptor can bind to many different ligands, or one ligand could bind to many receptors. So do genetic approaches help us address that in the way you're talking about with complementation?
Well, it does. I think first of all, the different ligands are usually in the same family may have very different functions. So, for example, we study quite a bit the ligands for the JAK-STAT pathway. I mean, here there is only one receptor for the different JAK-STAT ligands of the pathway, but there are three different ligands, and they are expressed at different times during development, and each of them actually have their own specific effect. So they all signal through the same receptor in different tissues, but if you mutate the ligands, you can see one of them is going to affect embryonic development. Another one is going to be acting as an hormone like leptin and so on. Another one is going to be involved more in proliferation. So we can really dissect the effect of each of those ligands by removing the receptor at the specific time points where those ligands are required. And then we can visualize the effect of the removal of the receptor in the target tissues at that specific time point. So you're correct that all those different ligands could be very complicated because they all act from the same receptor. But in general, at least in the fly, those different ligands are not really acting at the same time. So because we have ways to control very precisely when and where we can remove, let's say, the receptor activity, we can really visualize the phenotypes which correspond to the specific ligands.
Got it. Okay, that's very helpful.
Yeah.
Dr. Han, let's turn back to the question of AI and computation and how that fits in. So you talk about some of the strengths and limitations of that approach. So maybe we talked about one limitation in terms of glycosylation, but how about the strengths? Like where do you see AlphaFold and other structure prediction programs fitting into your workflow to map these ligand receptor networks?
Yeah, although I mentioned there's some kind of limitations of AI tools, I think it is still most powerful approaches to solve these questions. Let's say, for instance, if I perform the affinity pull-down mass spectrometry, then I would like to proceed with the in vivo validation. But before going on that, I think I can apply the AlphaFold screening. So with the hit identified from the affinity pull-down, and just to quickly check the binding score between the protein of interest and the candidate. And I can prioritize the few proteins to be tested in the in vivo screening. So I think still ready, and also there are a lot of biological findings have already published using the AlphaFold, especially in the extracellular protein-protein interaction field. So that is really powerful tool still. Yeah, I agree with that.
Okay, that's great. And I guess on the same vein, Professor Perrimon, you published the FlyPhone DB2 from your group, which used AlphaFold predicted protein-protein interactions integrated with, if I got it right, single cell data. So how should we think about that? Is integrating structure, computation, expression, and spatial data really the future of this field?
Yeah, so that's really another amazing tool, actually. The advances in single cell RNA-seq. I mean, especially in the flies, it's incredibly powerful because we can actually phenotype the entire fly by simply sequencing 20,000 to 40,000 cells. In fact, in the fly, we don't do single cell RNA-seq, we do single nuclei RNA-seq because there are a lot of cells which are multi-nucleated and so on. But yeah, so what we do is that if we are interested in trying to characterize a phenotype, so in the past, we would actually use a lot of different antibodies to look at different pathway activities and so on. But here, simply in one sequencing reaction, I mean, we can get the transcriptome of the entire body of the fly. And so we're able to build clusters about where all the genes, of all the different tissues. And then we can use, like, as you described, fly phone, which is basically a version of, so there are a number of different tools like this from a mammalian system called cell chat and a cell phone and so on. So we built the fly version of it, but you can simply use now those type of tools to try to make predictions about this ligand is expressed in this tissue, the receptor and some of the downstream targets are expressed in that tissue. So you can hypothesize that this ligand may be acting on that tissue. So you're able to build now a framework about all the potential ligand receptor interactions, assuming again that you know which receptor corresponds to which ligand, and then you can use that as a way to for discovery. But then you can test, you can try to validate some of those interactions. So I think it's really the way. I mean, where we're going now with this is by having tools to predict all the potential receptors of different ligands using AlphaFold multimer, and then combined by more experimental approaches to identify binders of specific ligands, putative receptors, together with gene expression on the spatial information using those UMAPs that you can generate with a single nuclear RNA-seq. Then you can really pretty quickly come up with a number of different ligand receptor interaction that you can validate in vivo using the tool that we have in flies to control spatial temporal activation or knockdown of all the different components. Because also one thing people need to realize, in flies we have genome-wide RNAi libraries of transgenic RNAi lines, which allow us to manipulate every gene anywhere, basically. All based on Gal4 UAS system, as well as we have libraries of every gene that have been targeted by RNAi, and the fly stocks are available in stock centers to allow those kind of in vivo validation. So we can go.
Right.
This is really the power of the fly, that we can, and now we can use those kind of very large scale approaches, integrate them, and then come down, select maybe 100 or 200 or so different putative interaction that we can validate using available tools for doing the in vivo analysis.
So that's really hugely powerful. Yeah,
yeah,
that's fantastic. I can see that integrating these different data sets is a great opportunity for computational biologists and just immensely powerful. So let me ask a different topic about translating these results to new medicines. A lot of listeners will be interested in drug discovery and therapeutics and new medicines for some of these really intractable diseases. So Dr. Han, let me ask you, when you think about deorphanizing these secreted proteins, where is the payoff in terms of drug discovery? Is it therapeutics directly? Is it target discovery? Or is it maybe understanding better the mechanisms for existing drugs or drugs already in development?
I'll say this review is focusing on the identifying the receptors of the certain secret proteins at this moment. But eventually, when we think about the FDA-approved drugs, this more than 70% of the drugs are targeting the GPCRs and extracellular proteins. So in that moment, in that case, I think adding another ligand receptor pair onto that the catalog is quite not just identifying the ligand receptors, but it is rather just expand the the market of the biopharm or the drug targeting the things. And also nowadays a lot of the biomedical scientists are interested in the identifying the biomarkers of the certain disease. So not only just a disease, just a aging condition, and also the recent paper published a lot of secret proteins has changed dramatically. So that could be something beyond that biology as well. So, but still, we don't know what the changes of the secret protein does in the aged conditions. So by doing the identifying the potential receptors of the novel secret proteins in certain disease condition or physiological condition can expand our understanding the underlying the biology as well, not only just the drug.
Right. That's a great point. Absolutely. All right. Well, let's wrap up with one more question for both of you. We'll go in turn, just thinking about students getting involved in secretome biology. So what advice would you have for a new graduate student or postdoc, somebody trainee interested in this area? What's the one thing that you would recommend that they do to have a kind of high impact in this field? Dr. Han, perhaps we'll start with you.
I think I'm also trainee, so but I'll say if I advise to myself like in back in like 4 years when I was a um fresh postdoc, I'll say just jump into the challenging project to solve the big questions. So do do the science that can impact more values. So for instance, I was thinking initially when I joined the Perrimon lab, I was thinking to just pick the one of the secret proteins and explore their unknown functions. But I think at the time I came to know that the tools to develop, I mean, to identify the protein-protein interaction is quite underdeveloped. So that's why I jump into this challenging project. I think I do have some progress, but it's still quite a slow progress. But still, I'm still confident to work on that. And I believe that is making some huge impact on the field. And also, my idea is also written in this review paper. So especially in the ligand receptor field, I think that that's the most biggest question and biggest the thing we need to take on right now.
Okay, that's great. Perfect. And Professor Perrimon, do you have any words of wisdom
Yeah.
for students interested in this area?
Well, I think what's important is to keep in mind the biological question eventually that you want to address. Because here, so what Hoon is doing is to try to develop a method because we saw a knowledge gap or in the way we are doing things. But eventually, and I keep asking him, so eventually, what would you focus on in terms of the biological question that you're going to be asking using the technology that you developed? And I think this is important to keep the question whether or not it's going to be, for example, interaction between the fly liver or the muscle and so on like this, because it really helps focus eventually the space of experiments and because you can do a lot of different things. So you really need to have eventually, you need to be able to say, oh, I'm interested, for example, in identifying the new ligands for GPCRs. So again, my advice is also not to lose sight of the biological question that you would address using the technology that you develop.
That's great advice from one of the leading biologists of our time, is always remember the biological question. I think that's perfect. So Professor Perrimon, Dr. Han, this has been a really insightful conversation. One of the things I take away is that we may finally be at a point where this deorphanization stops being a one-off, as we talked about in the beginning, and it really does become more systematic, telling us all what it means to be a living animal. So thank you both for your fantastic paper and for your research and for being here.
Thank you for the opportunity.
Thank you so much for this.
Great. The paper is called Approaches to Deorphanize Secretome by Myeonghoon Han and Norbert Perrimon. As a reminder, there's no cost to publish or read the articles in EXO Beyond the Cell. Thanks for listening to EXO Chats.